The script that's written in our genes directs us from behind the scenes. The words within it shape life's destiny. Hidden in your DNA is your genetic dossier. It tells your future and your history. How traits get passed from parents to a child is something that has kept us so beguiled. Cracking the code, genetic mysteries to unfold. Cracking the code, genetic secrets will be told. Cracking the code, genetic mysteries to unfold. Cracking the code, genetic secrets will be told. Cracking the code.
By the mid-1970s, scientists were able to extract DNA from any living creature and clone it within bacteria, a process known as genetic engineering. But they were still in the dark about the information within DNA, the long strings of chemical letters that determine our genetic destiny. Then, within a decade or so, scientists learned to manipulate DNA in amazing new ways, which allowed them to crack it open. They learned to find the variations in DNA that mark us as individuals, our DNA fingerprint, which revolutionized police work. They were able to read out the sequence itself, the four-letter software code that serves as the operating system of the cell. And using some ingenious new tools, they were able, for the first time ever, to hunt down disease-causing genes and pinpoint their mutations. But this amazing new DNA technology was in fact the result of combining together some quite simple tricks with DNA. The first such trick is to chop DNA into very precise bits. This is done with bacterial proteins called restriction enzymes, which are programmed to cut DNA in two wherever a particular short sequence of letters occurs. These recognition sites are unevenly distributed, so the resulting fragments differ in length. This symbol represents the simple trick of chopping DNA with a restriction enzyme. Hamilton Smith discovered the first restriction enzyme in the late 1960s.
My work was referred to at one point as a shot that was heard around the world.
And in 1978, it was rewarded with a Nobel Prize. Nearly 40 years after his landmark discovery, Hamilton Smith is still a hands-on scientist and still working with the enzymes that gave geneticists their first clue about the information hidden within DNA. In identical sequences of DNA, the same restriction enzyme will always produce identical sets of fragments. But in sequences that differ in the position of these recognition sites, the same enzyme will produce sets of fragments that also differ from each other. If these differences could be visualized, then scientists could use restriction enzymes to distinguish one piece of DNA from another, even though their sequences were still hidden away. This soon became possible thanks to a second simple trick with DNA, which is to separate out restriction fragments according to their size. In this case, the DNA fragments are tagged with a visible marker, so they can be followed. They are placed at the top of a slab of gel, into which they slowly sink.
The gel is a set of interlacing fibers. And DNA is a very large molecule, and it has to work its way through this kind of obstacle course of fibers.
Next, an electromagnetic charge is applied across the length of the gel. Since DNA has a negative charge, the fragments are drawn toward the positive pole at the bottom end of the gel.
The bigger the molecule, the more trouble it has getting through, and so it gets retarded, whereas the smaller molecules move more quickly. So you have a very powerful way of separating the different sizes.
Fragments of the same length travel down the gel at the same speed, forming bands.
And within an hour or so, you could see each of the different bands on the gel representing a different size fragment.
So using gel electrophoresis, a unique set of DNA fragments is converted into a unique pattern of bands. This symbol represents the simple trick of gel electrophoresis. Variation is essential to genetics. It was variation among pea plants that allowed Mendel to discover the laws of heredity, and variation among fruit flies that allowed Morgan to work out the first genetic map of a chromosome. Till now, geneticists had to rely on variation in phenotype, the physical expression of genes, to give them insight into the genes themselves. Now, by combining together these two simple tricks with DNA, scientists could study variation within the underlying DNA itself, the genotype.
It became apparent that this was going to be an incredible new tool.
One early and unexpected result of this new tool was the DNA fingerprint. In the early 1980s, Alec Jeffreys was a young genetic scientist at the University of Leicester in England. Like others, he was searching through human DNA for sites of variation. In one of their experiments, Jeffreys and his colleagues spread out in one lane of their gel all the restriction fragments from all the variable sites they could find in one human sample. They also did this in other lanes with other human samples.
So we had this one sheet of X-ray film, which had several unrelated people on it, and you could see how the pattern varied. Purely by chance, we happened to have a little family group, mother, father, and child, and you could see how the DNA fingerprint of the child was made up of a collection of half of the genetic markers in the father and half in the mother. Having got that first DNA fingerprint, I think one would have had to have been a complete idiot not to have spotted the applications. I mean, they just spoke out to us.
One obvious application was in crime solving. DNA left at a crime scene could be compared with the DNA of a suspect. This was soon put to the test in a high-profile murder case.
And this was actually a terrible case, local to Leicestershire, where I live. And it involved two young schoolgirls, both of whom had been raped and murdered. The first in 1983 and the second in 1986. Very shortly after the second murder, a young man acting suspiciously was arrested. In front of his family lawyer, gave a full confession to the second murder, but denied any knowledge of the first murder. Now, the police really had to tie this person to the first murder. The pattern of the two crimes was so similar, it really had to be the same individual.
By coincidence, Alec Jeffreys' lab was only 5 miles away from the first murder scene. The police approached him in the hope that his brand new discovery would help prove that both murders were committed by their suspect. Jeffreys and his team produced DNA fingerprints from semen samples left on both victims by their assailant. They compared them to each other and to the DNA fingerprint of the suspect.
Semen recovered from both of those victims was indeed from the same man, so the police were right. Both girls had been raped and therefore presumably murdered by the same individual. So that was no big surprise. The really big surprise was that the blood DNA that we had from the prime suspect, who had already confessed to one of the murders, absolutely mismatched the semen profiles taken from the two victims. That young man was released, and he was a very fortunate young man. Given his confession and some other circumstantial evidence, I have no doubt whatsoever that young man, without the DNA evidence that we provided, would have been found guilty of those crimes and would have been jailed for the rest of his life. The police were now put in the terrible position of having no prime suspect whatsoever, but having DNA profile information from the semen from the victims. And what they then did was something extraordinarily brave. They decided not to disbelieve in the technology, but to completely believe in it. They've got the DNA profile of the assailant. They were fairly sure that the assailant was a local man, local to 3 villages near Leicester. So what they then asked was then initiated was the world's first DNA-based manhunt.
Double murder hunt, police to blood test 6,000 men to flush out the killer.
They asked men between, I think it was the age of 17 and 34, which is the most likely age bracket for a sex attacker, to voluntarily come forward, give a blood sample, and then that would then be tested to exclude them from matching the crime scene samples. The police then went through that massive hunt, and they failed to identify anybody that matched the crime scene sample. And the lead went cold, and everybody started getting very worried that here was a technology that wasn't delivering the goods. Then the police had a lucky break. There was a woman at a local pub was sitting there overhearing or listening into a conversation, and there was a London conversation. Then a man started talking about this case and said, oh, incidentally, he stood in for someone else, giving a blood sample. Now, this woman was a little bit worried about this. She worried about it for a while, and she went to the police and said that I've heard that there's this man who stood in, given a blood sample instead of someone else. Perhaps you ought to look at it. The police then checked up on this person, interviewed him, found out who he had substituted for. The person he substituted for had the right profile in terms of a criminal record of being the true perpetrator. That man was then arrested. At the point of arrest, he completely confessed on the spot to both murders. He was DNA typed, he matched the crime scene samples. The police have finally got their man. You needed the DNA evidence, but you needed good classic police work as well. And it was that combination of the two that flushed out the true assailant. And I think without that combination, he would have gone uncaught for a long time and unquestionably would have killed again. He was a serial murderer. So it was a quite extraordinary case for DNA to cut its teeth on. That's where it all started.
By the mid-1970s, geneticists had learned to analyze DNA by studying its fragments. But the information within DNA was still hidden away. Then this message suddenly became visible thanks to another amazing new technology, which turns DNA fragments into DNA letters. Once again, this was the result of putting together some much simpler tricks. One such trick is to use heat to split apart the 2 strands of the double helix.
The forces which hold the 2 strands together are relatively weak, and you can actually pull those 2 strands apart by heating a solution of DNA up close to boiling point.
This symbol represents the simple trick of splitting apart the double helix. Another simple trick is to use a single strand of DNA as a mold or template to create its complementary strand, using single nucleotides as building blocks. They attach themselves according to the base pairing rules, A's bonding with T's and C's bonding with G's. Then they are fused together into a new strand by an enzyme called DNA polymerase.
They're there as part of our normal machinery for making more DNA. As 1 cell replicates to be 2 cells and 4 cells and 8 cells, we always have to double the DNA in doing that. And so we've been able to extract those enzymes from living tissue and then use them as laboratory tools later on.
This symbol represents the simple trick of DNA polymerase. Fred Sanger, an English biochemist and prior Nobel laureate, used these 2 simple tricks with a chemical twist thrown in to read out a DNA sequence. First, he separated the 2 strands in multiple copies of the fragment he wished to sequence. He then used the copies of 1 strand as templates upon which to grow new complementary strands. But mixed in with the normal building blocks were some altered A's. Part of their backbone section was missing. So when one of these altered A's happened to bond to a T, the new strand came to a halt. Each such A also carried a radioactive marker. With enough time and templates, an altered A bonds with every T in the target strand, producing complementary strands of varying length, but all of which end with a radioactive A. Everything is then split apart, producing a set of radioactive DNA fragments that can be sorted out according to size and then revealed as bands on an X-ray film. Since each fragment ends in an A, The relative position of the bands corresponds to the relative position of every A in the complete sequence. Sanger repeated this process, this time using an altered G instead of an altered A. These bands revealed the position of every G in the sequence. In similar fashion, he filled in the position of every C and then every T. He was now able to read off the complete sequence of the complementary strand. It was a trivial matter to then use the base pairing rules to work out the original target strand. Word of this breakthrough soon reached Hamilton Smith, who was one of the first to put it to the test.
And it was just incredible. I could read 50 bases. I mean, for the first time, almost the first time in history, you were seeing the sequence of a gene. It was an incredible experience. I rushed out with the dripping X-ray film, and everybody gathered around and we looked at it. We were in awe, you know, that you could take somebody off the street and they could literally read off the sequence. It was incredible. In a period of 3 weeks, we sequenced 800 base pairs and obtained most of the sequence of one gene, which we published a few months later. It was incredible.
In 1980, Fred Sanger won his second Nobel Prize, this time for the invention of DNA sequencing. The Sanger method remains the basis of today's high-speed automatic sequencing machines, which can read out more than 1 million DNA letters a day. DNA sequencing soon led to another amazing new DNA technology. Each of these small machines is a powerful DNA factory that can fish out from the vastness of all our DNA any particular sequence of interest, such as a gene, and then multiply or amplify it into literally billions of copies, all within a few hours. This technology is called the polymerase chain reaction, or PCR for short, and in the late 1980s it revolutionized genetics.
PCR, or DNA amplification, I mean, is an extraordinary technology. I mean, it's one of these technologies I wish I'd thought up.
Like DNA sequencing, PCR makes use of the two simple tricks of splitting the double helix into 2 strands and then using a single strand and DNA polymerase to create a new complementary strand. But in between comes another simple trick, which is the key to its power. It uses a short strand of nucleotides as a kind of homing device or probe to seek out a complementary sequence to which it can bind, according to the base pairing rules. The mating together of 2 complementary sequences is called DNA hybridization. This symbol represents the simple trick of hybridization. PCR makes use of this trick to pick out the target sequence from all the other DNA in the cell. After the double helix is split apart, 2 custom-made probes called primers are introduced. One primer hybridizes to one end of the target sequence on one strand, and the other to the opposite end on the other strand. These then become the starting points for 2 new strands, which are fused together by DNA polymerase. When this cycle is repeated, more new strands are created, some of which are exact copies of the target DNA. With every cycle that follows, these copies double in number.
And in no time at all, you got yourself 1 million copies or 1 billion copies of your chosen favorite piece of DNA.
Instead of having to clip out a piece of DNA with a restriction enzyme, you could simply, using the lab jargon, you could lift out that section of the DNA by simply amplifying it. It literally gave us 1 billion-fold greater power than we had before that time.
In 1993, a Nobel Prize for Chemistry was awarded to Kary Mullis, the American scientist who invented PCR.
That DNA sure does amaze. Come on, everybody, let's sing its praise. Oh, you can break it down, a DNA, and build it up again. A DNA, a deoxyribonucleic acids now in play. You can hybridize or synthesize it. You can do so much, it's no surprise. It's worth a mint. It's a fingerprint. That's scandalizing, tantalizing DNA. DNA. Yes, DNA is made for manipulation. What do you say, OJ? You can chop it up and spread it on a gel. On a gel, that's how. Well, that's how we learn to search out variation. Riff, lip, yeah. From where those fragments dwell, there's a lot that you can tell. Oh, you can read it out. A DNA and see what it's about. A DNA, a deoxyribonucleic acid's on display. It's on display, yeah. A chain reaction multiplies it. You can use some probes to analyze it in a chip at quite a clip. That hybridizing, hypnotizing DNA, commercializing, mesmerizing DNA, revolutionizing, so surprising DNA.
Geneticists finally had the weapons they needed to hunt down the faulty genes that cause simple genetic diseases, something they couldn't do before. The first step in any gene hunt is to narrow down the search to a small area within a single chromosome. Until now, this was done by linking the unknown gene to another gene whose position had already been marked on a genetic map. This identifies the underlying bit of chromosome in which the unknown gene must be located. The concept of genetic linkage was first developed almost 100 years ago by Thomas Hunt Morgan and his students using fruit flies. They studied simple inherited traits that are polymorphic, meaning each trait can take more than one form. They realized that when two unusual forms coexist in the same fly and then travel together down the generations, the two underlying genes must be close to each other on the same chromosome. After discovering a number of gene pairs linked together in this fashion, they did crossbreeding experiments to measure the strength of these linkages. These numbers told them how far apart one gene must be from the other. It's a strategy that worked well in lab animals like fruit flies, but not so well in humans. We are complex creatures in whom it is hard to find the simple polymorphic traits that can serve as genetic markers. What's more, the crossbreeding experiments needed to map these markers are impossible to do in humans. Then around 1980, geneticists realized that these new simple tricks with DNA had opened the door to a rich new source of human genetic markers. Our linear four-letter scripts are largely the same, but periodically the letters vary slightly from person to person. This often occurs at points where the same few letters are repeated over and over again. These repeats often show polymorphism in their length, and so too do the larger restriction fragments that contain them, which meant these DNA polymorphisms could be detected using the simple tricks of creating restriction fragments and then spreading them on a gel. This became even easier after PCR allowed geneticists to fish out and amplify any particular fragment they needed. And because these DNA markers are present in all of us, linkages can be established simply by observing which DNA variations travel together down the same family tree, which did away with the need for cross-breeding experiments. Even mapping DNA markers onto a chromosome was now much easier. Labeled DNA probes can be made from these markers and then directed at the chromosomes themselves. Each probe will hybridize with its marker, thereby revealing its chromosomal location. So it was now theoretically possible to track down any simple disease-causing gene about which nothing else was known using DNA markers only. This theory was soon put to the test with the most common of the lethal genetic diseases, cystic fibrosis. Its primary effect is in the lungs, leading to increasing shortness of breath and eventually an early death. From its pattern of inheritance, it is clearly the result of a mutation in a single gene. But there were no clues as to its whereabouts. So a number of scientists decided to use this new DNA-based strategy to hunt down the cystic fibrosis gene, in the hope that finding it would lead to a better understanding of this important genetic disease. In 1985, Lap Chi Choi, a scientist at Toronto's Hospital for Sick Children, made the first breakthrough. He found a DNA marker on chromosome 7 that was linked to the disease.
And then once that news broke out, other people had markers on chromosome 7, test the linkage, and narrow the gene down to a smaller region very, very quickly. And from there on, it was everybody's guess.
This region was flanked by 2 markers, about 1.5 million letters apart. Next came an even bigger challenge, rummaging through all this unsequenced DNA in search of an unknown gene.
We had no idea because it's really something what we call uncharted territory.
The method they used is called gene cloning. First, the region is broken up into small bits of DNA, each of which is cloned in a bacterial culture. Then, starting at one end, a clone-by-clone search through all this DNA begins.
This is what we call chromosome walking. We isolate pieces of DNA successively, one at a time, like walking, a step at a time.
But Sheva Karam was the team member in charge of guiding this walk. She is now at Hebrew University in Jerusalem.
And in each such step, we looked for polymorphic markers, which showed us if we are getting closer or are getting to the other direction.
After further narrowing the search to a small area within a single clone, the next step was to painstakingly search out every gene within it.
At the beginning, nobody believed that this will work.
We were talking about maybe 200 genes in that region. We had to go through the whole region systematically to look at the gene one at a time. You don't want to miss anyone along the way.
And each gene we found, we tested whether it could be the CF gene itself.
Francis Collins, then at the University of Michigan, helped out during this very difficult phase.
You had a lead, half the time it turned out to be wrong. People got burned out and disgusted and just left and did something else.
In the meantime, at St. Mary's Hospital in London, England, a rival team was hunting for the CF gene in the same small area. It was headed by Bob Williamson.
It was a very close race. With poker faces we met, and they tried not to tell the other group the real data.
In the summer of 1988, the 2 rivals shared a seminar in Toronto, but little else.
We had a lot of problem walking because it just stopped. That's right.
Williamson was hiding a bombshell that he was about to drop. His group soon published a scientific paper in which they claimed they had found the cystic fibrosis gene. But it turned out to be a false alarm, as their Toronto rival was quick to point out.
He did not show, did not give very good evidence that the gene he has got in his hand was the right gene. Put your hand.
A year later, it was the Toronto team's turn to celebrate. This time it was for real. They had beaten their rivals to the finish line, but not by much.
And it turned out that they were very, very close. Actually, they were within the gene, but they were not correctly oriented at the time, so they didn't know that they are within the gene.
Effectively, it took 10 years, most of the decade of the 1980s, to find this single gene with its common mistake, which is a 3-letter deletion, which is the common cause of cystic fibrosis.
This historic discovery, using some new tricks with DNA, made headlines around the world and inspired the next new era in genetics.
It was an example that everybody else said, wow, this could be done.
Cracking the code. Genetic futures will be foretold. Cracking the code. Genetic power about to explode, cracking the code. Genetic mysteries to unfold, cracking the code. Genetic secrets will be told, cracking the code.